So our next speaker is Professor Mohit Kumar Jali from IISc Bangalore, and he is going to talk about design principles of large cell-fed decision-making regulatory networks today.
Thank you very much. I would like to begin with thanking the organizers for giving this opportunity to present some of our work here. What I'll talk about today is looking at some different regulatory networks implicated in diverse biological systems and try to understand what are some of the organizing principles that underlie and control the dynamics of those networks, which eventually impact how a cell behaves and how an organism behaves. So cellular decision making, the concept here is that cells in our body are constantly being exposed to a large variety of signals which vary both in space as well as time. And cells themselves have an intricate machinery of all these different biomolecules interacting with each other. Cells in a given population can respond very differently to the same amount of signal that is being given. So there is this elements of heterogeneity that are present or variability that are present. And how exactly a cell responds to a given signal is the eventual outcome of all these different interactions and the emergent outcome of these biochemical and biophysical interactions that are going on operating across different length and time scales. So this interest of this topic of cellular decision making has interested physicists for a very long period of time. One of the phenomena that has been studied for more than 50 years now comes from that of bacteriophage lambda. So in this particular scenario, you have a phage. Phage is something that infects, it's a virus that infects E. coli bacteria, and then once this enters this particular bacteria, either it can just lyse the cell, kill the cell, and create many more copies of it, or it can also get stably integrated into the genome of the bacterium cell. And this decision, what is called lysis versus lysogeny decision making, has been studied all the way from 1950s, and more molecular information has been created, therefore enabling us to create more accurate models of how different factors influence this. So of course, I'll not go into the details of this. I'll present one another example. This is a paper 30 years ago, that of cell cycle, which is also one very well characterized and studied mechanism across different biological systems. So here is a set of different chemical reactions representing different biomolecular scenarios. They interact with each other, which you can then condense to write some sort of coupled differential equations, solve the emergent dynamics of it. So in this particular scenario, you can see that there are oscillations which correspond to the response of cell going through a cell cycle. Of course, a cell can be arrested and not go through a cell cycle, so that's a stable phase and other scenarios. These responses are not always deterministic. So even if you know all the biochemical reactions, you know all the variables, there is a lot of stochasticity that goes into it. So for instance, this beautiful experiment done almost 20 years ago now, what they did was that in a bacterial cell population, they tagged two genes, one with a red fluorescent protein, one with a green fluorescent protein, and then measured the dynamics as a function of time. So what you can see is that if these dynamics were absolutely correlated, if the rate of production of these two genes was identical as a function of time, then what you would have seen is that the green and yellow fluorescence protein have the exact same intensity, and what you see is more like a combined signal. But what was seen is that different cells actually gave rise to varying degrees of red and green despite having everything identical. So this is not just a genetic mechanism. There are all these stochastic biomolecular reactions that are going on because of limited number of molecules that we are talking about, which can also change the behavior of a cell. You can also have other scenarios where, let's say this is an experiment done in multiple cancer cell lines over 10 years ago, where you see that in a so-called genetically identical population, there were cells of different phenotypes. When you segregate them and culture them independently, you see that all of them come back to a very similar distribution, similar to what they started from in the parental population. So this was then fit with a Markov model, figuring out the rates of transition over much longer timescales. So that's all about what is going on at different scales. Now, if we look at, we started with what is going on at the overall cell level. at a population level. Now let's see what's going on within a cell. So within a cell, again, this idea of landscape or Waddington lands, which was mentioned by Waddington in 1940s, therefore called as Waddington landscape. What this represents is that when there is a cell which can give rise to multiple different cell types as it happens in the development of an organism, the choices that this cell makes is represented through this scenario where once it goes down, it cannot go up. That's the projection, that's the representation of this. And every time this ball rolls down, there is further and further canalization happening. There is further and further limited repertoire of cells that it can give rise to. Now, what controls the shape? What controls how many valleys exist? What controls what's the depth of each valley and so on? Well, of course, in 1940s, we did not have molecular biology to that extent, and we did not know the interactions between these molecules to that extent that we can actually map this out. But 80 years later, today, we have that kind of information that we can begin to map these landscapes in a more quantitative and predictive manner. But one general feature that you'll see in some other examples I'll talk about today is that of multistability. That many of these networks are multistable in nature. They can give rise to cells being able to take one of the multiple possible phenotypes, multiple possible states, and because it is multi-stable scenario and there is this constant noise due to multiple different reasons, low molecules, a low number of molecules, asymmetry in cell division, etc., these cells can switch back and forth between these different phenotypes. So even the heterogeneity profile that you see at one time point can be very different than what you see at a later time point. So here in this case, what you see is there are two molecules which inhibit each other and activate themselves. Inhibit means that the rate of reaction goes down, activation means that the rate of reaction goes up. And in this scenario, these two stable states come in, mentioned segregated by the separatics of the unstable state. Okay, and this exact same network that I presented to you earlier was also one of the first networks which was synthetically constructed 20 years ago. So going back to Feynman's statement, what I cannot create, I do not understand. So this network was actually created. It was inserted in bacterial cells. This is the structure here, which you can mathematically represent as two different molecules, U and V. V is repressing the production of U, and U is repressing the production of V. And then you have the usual degradation and the usual production rates. What you see here in these phase diagrams is that this particular system can give rise to three scenarios. A monostable state in which U is high and V is low. Another monostable state in which V is high and U is low. And in between, you have this bistable scenario in which both of these states can coexist. Now, why these two states exist, U high, V low, or V high, U low, is very simple because they are inhibiting each other. So if U is high, it will always continue to repress V so that V does not go high enough to repress U. Or similarly, if V is high, then it will continue to repress U such that U cannot go up to repress V. It's like a seesaw. It's like two people who hate each other and do not want to go to the same event. So eventually only one of them ends up going there. U, V are these... I'll come to that. Yeah, that's the effective potential in this particular scenario. Yeah. What are the absolute values? We'll come to that. So I'm so far describing it in a very qualitative binary kind of way. We'll come to the more quantitative ways in a minute. Okay. So this system, you can draw the null lines for this for the kind of equation that we talked about, or also try to understand this graphically. So this is the response of how when B increases, A decreases. This is the response, the inverse of it when you're talking about the other link in the network here. You can do the stability analysis, figure out which is stable, which is unstable, and these two states end up being stable, and that is why it's called as a bistable system. This is the more generic form of the equations you can write down. You can introduce a little bit more complexity by adding more variables into the system as we were looking at the previous cases. But the general formulation stays the same. There is a production, there is a first order degradation, and then there is some nonlinear regulation that is going on here. Okay, now why should we study this particular network of two molecules inhibiting each other? because this has been seen at multiple different points whenever cells have to take a decision. So go back to the landscape, the Waddington landscape, when there are more and more bifurcations, more and more cell types being created in our body. Life starts with a single cell, and we have approximately 200 to 250 cell types eventually in a fully developed organism. So at all those places, what has been seen, or not all, but many of those places, what has been seen is that there are two molecules which are involved in the exact same formulation. Two of them will repress each other, therefore enabling the cells to go in one trajectory and therefore restricting its ability to go to the other one, right? So it's a progressive process that continues to happen. But clearly, as we were talking about, networks are not just two molecules inhibiting each other. They're embedded in much larger repertoire of signals, and that is affecting their ability to give rise to that clean bistable behavior. So what we are going to talk about today is that if you look at much larger networks, do they also show multistability in the first place? And if they do, then why are those networks designed the way they are? Right? So we are going to move from looking from simple two-node inhibiting each other to a much larger network going all the way up to approximately 80 nodes, 200 edges size networks, and see what the dynamics is. And is there some underlying principle that we can expect to arrive at by looking at those dynamics? Okay, so the system I'm going to focus on today is something that is the major direction in our lab, how cancer cells metastasize, how cancer cells move from one organ to another. Now, during this process, where do state transitions come in? I'll just explain that in a minute. So metastasis remains the major cause of all cancer-related deaths. There are no unique mutational signatures that have yet been identified for metastasis. Largely happens through this fantastic freeway setup in our body called blood vessels. But it's a highly, highly inefficient process. 99.9% of the cells that leave the primary tumor are not able to grow metastasis. Imagine what could happen if this process was more efficient. The reason why this process is so inefficient is because cells are constantly facing a very diverse set of challenges. So think of a cancer cell which leaves the primary tumor, is in the bloodstream somewhere. The environment around it is constantly changing. Oxygen levels are changing, nutrient levels are changing, immune cells are attacking it, there is shear stress being applied by the blood, etc., etc. So how does this poor cell then navigate through all these challenges and then eventually end up in a completely different environment, which it has? So if once a prostate cancer cell ends up in bone, it has no idea how to survive in that environment. So it has to adapt to that environment as well and eventually colonize it. Right? So there are all these different challenges that this cell has to face, and that is what these bottlenecks put together cumulatively is what reduce the efficiency of this process dramatically. So therefore, what they need to do is to adapt fast and to adapt reversibly. And mutations are neither fast nor reversible. They are slow processes and they are not. So the phenomena that we have been looking at is something very well established in the context of metastasis, is called as epithelial to mesenchymal transition. So epithelial cells are those cells in the body which do not have the innate ability to start moving and migrating and invading. These are cells which are stuck in a specific 3D geometry very tightly with their neighbors in a specific polarity. Think of prostate cancer, breast cancer, liver cancer, lung cancer, etc. All of these are epithelial organs, and these cells are epithelial cancer cells. Now, when they have to move, they have to break their bonds with their neighbors and gain the ability to migrate and invade, which is the property of mesenchymal cells in the body. And once they reach the other organ, they have to again leave the property of migration and invasion and regain the property of adhering to their neighbors and form a tumor. And this is the state transition that we will be talking about. So there is EMT going from E to M, and then there is MET. EMT triggers the first step of metastasis, which is cells leaving the primary tumor, and MET is the last step of process when cells have to settle down in a different organ. And if you look at how people in the field have been thinking about EMT-MET dynamics, it has been implicated as a major process in metastasis. But in all of these scenarios, in all these diagrammatic representations, one thing which stands out is that EMT has been thought of as a very binary process. Cells are either epithelial or mesenchymal. So that was the first question we started with, was that is EMT truly a binary process, or are there cell states somewhere in between in addition to those extreme epithelial and mesenchymal states? So what we had done earlier was we identified a regulatory network controlling EMT and converted these regulations into a set of differential equations, solve these equations. When you look at the bifurcation diagram, here the x-axis is some EMT inducing signal and the y-axis is the measure of EMT-ness. What you see are three different states: epithelial with the lowest levels of Zeb of this molecule, mesenchymal with the high levels of Zeb, and another state which has intermediate levels of Zeb. So our model predicted that EMT is not a binary process, and you can have these cells exist in a hybrid epithelial mesenchymal state. Then we collaborated with a set of lung cancer clinicians to check whether individual cells in a population can co-express epithelial and mesenchymal molecules together, and can they do that over multiple time points, not just at one time point. And this is what the diagram shows here. Red and green represent two different molecules, one belonging to an epithelial behavior, the other belonging to a mesenchymal behavior, and they continue to do that over multiple passages in the lab. There have been various other models that have come up in the past 10 years since we started working on EMT, and each of them takes a slightly different network, takes a slightly different modeling strategy. Some of them are ODE based, some of them are discrete Boolean logic based models. But all of them, as you can clearly see, do indicate that there is one or more of these hybrid states that exist. So now there is enough evidence also experimentally in the literature that has been collected saying that epithelial to mesenchymal is more of a spectrum where there are these multiple states that exist. Not only these states exist, but also these states are in fact essential for metastasis. These states are in fact the most aggressive ones and the most fit for the process of metastasis. There is also clinical data supporting the same thing. So for instance, here, what you see in this graph, the x-axis are the 4 different types of breast cancer. TN stands for triple negative, which is the highest, which is the most aggressive one, and it has the highest levels of these hybrid cells. Also, there has been data collected across different cancers now over past 10 years or more, saying that even if you have a small proportion of these hybrid cells, that can be sufficient to give a much worse outcome to the patient. So the focus of today's talk is to further characterize where these hybrid states come from and what properties of the underlying networks that control EMT make hybrid cells more aggressive. Okay. So there's been extensive experimental data showing that while epithelial and mesenchymal states are more stable relatively, the hybrid states are much less stable. The experiments done are the following. You start with a set of epithelial cells, don't give them any induction, just let them grow in the lab. Come back after 2 weeks, 2 months, you will still see most of those cells are epithelial. You do the same for mesenchymal, you see a similar behavior. But if you do this for hybrid, most of them within 2 to 3 weeks have lost their hybrid properties and have switched to either epithelial or mesenchymal. So this has been consistently seen in vitro, this has been seen in vivo as well. So the questions we were interested in is why? Why are so many studies reporting such a consistent pattern? This study was led by my PhD student, Kishore. So what we did here is now look at much larger networks, which we have not put together these networks. These are from various different groups working on EMT modeling. What you see in each of these networks is that, let's focus on this one for instance, each red node is an epithelial player. We know that from experimental data that when levels of these red players is high, it's an epithelial state. The blue ones are all mesenchymal state. The gray ones are those nodes in the network which are not involved in any feedback loop. So either they are inputs or they are outputs. They are not affecting the network dynamics dramatically. Red node is inhibition, red arrow is inhibition, and green arrow is activation. So when you look at these networks, and the way we have simulated these networks is Boolean dynamics, using asynchronous updates. So at a given time step, all we are counting is how many edges are coming to a node and whether the number of activating edges is more than the number of inhibiting edges. If yes, you turn on that node. If no, then you turn off that node. It's very simple, no parameters, no kinetic parameters. Okay. So when we simulate this particular network over a large number of initial conditions, what you see is that epithelial and mesenchymal states, which means epithelial states means all of these nodes are on and all of these mesenchymal nodes are off. And mesenchymal state is defined the exact opposite way. All of these nodes are off and all of these nodes are on. They dominate the landscape. You get much more epithelial and mesenchymal states as compared to the hybrid states. So their frequency is less. You can quantify this for all different networks, and you see that with the exception of this last one, epithelial and mesenchymal seem to dominate the landscape, and the hybrid cells, you don't actually get as many of them. Then you can ask the question, how do we know that this outcome is specific to this network? So you take this network and you shuffle the edges in this network, therefore generating many non-biological hypothetical networks. And for each of that hypothetical network, you again do the simulation and ask how many steady states exist. As you can clearly see in this diagram, in this histogram, for 1,000 different random networks that we simulated, the wild type network or the biological network has among the least number of steady states that these networks would allow for. So this means that this is something very specific to this particular set of, this particular biological network. You can quantify this in many more ways. So here what we have done is define something as coherence. Coherence means that when you take a steady state, then you flip the node, one of those nodes at a time. So you turn one of the nodes which was on to off or off to on and see whether the system comes back or the system disappears goes to another steady state. So for instance, in this case, if you have all these 7 nodes, you flip them one by one. In 4 of them, the system goes to another state. In 3 of them, the system comes back. So coherence is 3/7 on a scale of 0 to 1. For all the 5 networks that you see a very bimodal distribution. So there is one set of states which has low coherence and another set of states which has high coherence. You can define, going back to the spin glass kind of models, you can define something called as frustration. Frustration here means that whether a given edge obeys, whether a given state obeys a given edge or not. So if A is activating B and both of them are on, it is not a frustrated node. But if A is activating B, A is on and B is off, you have a highly frustrated setup. So you can again quantify this, how many frustrated edges are there as a function of total number of edges for all different 5 networks. You see a very clear pattern. Again, the more the coherence, the less the frustration. And the less the coherence, the more the frustration. They are strongly negatively correlated properties. So now you have steady state frequency, you have coherence, and you have frustration. Let's put all of them together and see what we get. We see these 2 types of states. One state which has very high frequency, very less frustration, and very high coherence. These we found were all epithelial or mesenchymal states. The other set of states, which when we look more detailed into that, are hybrid states, which have much less frequency, much high frustration, and much less coherence, the exact opposite properties. Okay? So then this tells us that hybrid states are frustrated, and probably that is the reason they are more plastic, because they are not obeying the rules of the network. So they need a little bit of perturbation to get out of the relatively shallower valley and then become epithelial or mesenchymal. But the interesting question then comes, why? Why are hybrid cells more frustrated? Is there something inherent in the network topology itself that makes them less frustrated, that makes them more frustrated? Okay. So then what we did is we look at this network. You can convert this network into a matrix where each row is the source, each column is the target of a given edge, and wherever there is an edge, whether it's an activatory or inhibitory edge, it can be red or blue respectively. When there is no interaction, it's just an empty space. So you can see it's a very sparse matrix because in a 22 cross 22 matrix, you have only 82 interactions. Only 20% of this is filled. Now, this is for direct interactions. Node A activating or inhibiting node B. But in real biological systems, you can have the signal coming in indirectly as well. So A can activate B, which can repress C, that can activate D, which can activate E, right? So there can be this propagation of signal that can also happen. And the longer the path length, the less robust is the propagation of the signal, right? So what we did is we multiplied this matrix by itself to capture for those indirect interactions and penalized it for the length of the path that we were looking at. What you see then is a very clear pattern. This is what we call an influence matrix because now we are not just looking at the adjacent interactions but also the indirect interactions. So now you see that these set of molecules are all effectively activating each other. These sets of molecules are also effectively activating each other. But when you look at any interaction going from any of these molecules to any of the other molecules, it's inhibitory. And similarly, if you look at any interaction going from any of these sets of molecules to any of the other one, that's also inhibitory. Okay? You can quantify the team strength on a scale of 0 to 1. You can do the same experiment, randomize biological networks and ask whether this formation of teams is something specific to biological networks or seen otherwise. The histogram clearly tells you the answer. 99.9 percentile is where the biological network ends up, irrespective of which of the 5 networks you're looking at. Okay, so this is very specific to the biological network. So then the question comes, how are teams and frustration connected? We see these teams, we see there is frustration, but are these two ideas connected to each other? So then we did these correlations across all the 5 different sizes of networks, where we correlate how we correlate with the group strength. So what you see is that if you look at the minimally frustrated state of enabled by any given network, that correlates strongly negatively with the team strength, telling us that the stronger the teams are, the more likely you are to end up in a less frustrated state. Okay? You can do this further across all these networks. So that gave us this hypothesis that teams may be supporting only epithelial and mesenchymal phenotypes, but not the hybrid phenotypes. The hybrid phenotypes, poor guys, are left on their own. They have no team to support them, and therefore, when you give them a fluctuation, they disappear. But epithelial and mesenchymal have a lot of backup available to them and interactions which pull them back into the same attractor or into the same valley. So we did this analysis. Here in this diagram, what you see is the x-axis, instead of changing one node, now we are changing many, many nodes at a time. And you start from either epithelial, mesenchymal, or hybrid. The color tells you when you have completely exited that particular set of attractors. If you start from epithelial or mesenchymal, you need approximately 40 to 50% of nodes to be flipped so that you can switch your state. When you start from hybrid, as long as you have switched only 10% of the states on to off or on off to on, 10% of the nodes, sorry, you have begun the process of them exiting that attractor and going somewhere else. Right? So it's a much shallower basin of attraction that these have. Sorry. You can look at the dynamics further. If you start from an epithelial state, until 25% of the nodes are being perturbed, the cell stays epithelial. When all of those nodes are flipped, the cell becomes mesenchymal. In between, what you see is that some cells are still epithelial, some are mesenchymal, which is because there is multi-stability in the network. So even if you have given a perturbation, some of them can come back and some of them can disappear. But if you do the same for a random network, for a non-biological network, which does not have teams, you see a very scattered behavior, and you don't see this sigmoidal curve. You don't see that initially there is a resistance for the cell to exit. Then there is an almost linear behavior, and then there is a saturation. What you see is more of a logit curve, which basically tells you that the more you perturb, the more further away you get. It's a much more continuous process rather than this threshold-like behavior. You can then delete these edges and then see how the group strength decreases, how frustration increases, and so on. So what we are saying here is that if you have strong teams, then you end up in a scenario where there are two deep attractors, an epithelial and a mesenchymal one. And if you have weak teams, then you have much shallower basins of attraction, and that allows the cells to switch back and forth much more easily. That allows more heterogeneity, and we have seen some unpublished clinical data that allows for more metastasis as well. This is not something true to just to EMT. You can look at other scenarios. Here we looked at in melanoma or in small cell lung cancer. We again see the formation of teams. This formation of these teams is specific to these biological networks. If you perturb the network, you don't get teams. And you see the similar behavior that the higher the team strength, the higher the coherence and so on. Okay. So I'll quickly talk about two other scenarios where we saw these teams and explored this, why these teams exist in a little bit more detail. One of them is this highly aggressive cancer called as small cell lung cancer. There is only 7% patients are able to survive in a period of 5 years after being diagnosed. There's patients already have metastasis by the time they get diagnosed, and there is no targeted therapy available for this cancer. Okay. There's a network which was available for this particular cancer, and the questions we started asking is what networks underlies multistability, if there is multistability in this, and what features of topology enable this multistability. So this work was led by two undergraduate students at IISc, Lakshya and Uday, majoring in physics and biology respectively, and now also together with Kishore. So what we found was that in that large network, once you simulate the dynamics of that network, you see predominantly only 4 states which sum up to give approximately 99.5% of the frequency of those states. And all those other states were much less frequent. Here, a filled box means that a node is on, and a white box means that a node is off. You can again ask the same question. You can do edge swapping and generate many random networks. What you see here is that the biological network had only 4 states, but on average, the random networks have anywhere on the order of 10,000 to 1,000,000 states. So the biological network is really constraining the phenotype space, multiple orders of magnitude, all the way from 5 to 6 to that of only 4 states. Right? And none of these 1,000 networks gave rise to any of these 4 states. So these are completely disjoint sets of solutions that we are talking about right now. Okay? Now this is the influence matrix for that network. So here are these 22 molecules that activate each other, these 11 molecules which also activate each other, and these 11 inhibit these 22, and these 22 inhibit these 11. Okay? Now this is to us one of the most exciting things, that you can actually predict the most frequent solutions of a network even without simulating the network. If you have the influence matrix, we found that within an error of 1 to 2%, we were able to tell what the most frequent solution of that interaction is going to be. That saves us computational time a whole lot. This is what our model predicted, that these sets of molecules are correlated with each other, and they form these 2 teams. This is what the experimental data also shows up. So whatever we are seeing in the model can directly be validated using data from cell lines, using data from patients, and so on. Okay. So in this case, we were able to reduce this complex network to a 4-node network because there is so much redundancy, and there is this formation of those 2 teams which are basically inhibiting each other. Once you simulate this smaller network, you get the exact same outcome as you were seeing from that larger network, and these 4 phenotypes then you can also experimentally validate. Another instance, melanoma, which is not an epithelial cancer but highly aggressive cancer again, where we know that cells can exist in multiple states. Malvika, who led this work, was an undergrad student in the group. She was able to identify a network which could enable these 4 phenotypes. She also came out with this existence of teams. So there are these sets of players which are all positively correlated with each other, another set of players here, and then they form these teams here. And here what we also saw was that there were 2 teams which were classifying 2 behaviors, and then if you go deeper, there was another set of teams which was present which could bifurcate the behavior. Using these teams and these landscapes, we could also explain experimental data. So for instance, in this case, we know that when you take melanoma cells and treat them with a given drug, over a long time scale of approximately 2.5 months, you see that some cells die, but some cells switch to another state, which is drug resistant, and then they stay there. Okay? So the drug, there is drug-induced transition which is also going on in the system. So the drug is having an impact in terms of killing the cells, but it also has a side effect in terms of increasing the fitness of the system. So we were able to explain the frequency of these states. We could also look at high throughput transcriptomic data that is becoming more prevalent these days and identify where exactly is this bifurcation of cell state trajectories happening, ones which are dying and ones which are switching to a completely different state. And then you can, going back to Partho's question, you can here construct landscapes. The landscapes here, I mean, this is a non-equilibrium system, so you can't define the exact potential for them, but you can define pseudo potential for these by looking at the negative log probability of where exactly these states end up. And you can change the landscape, you can change how many states exist, you can change their relative stability, and so on and so forth by changing various different nodes in the network. Okay, so I'll just summarize this part. So we have seen the existence of teams in multiple different behaviors. And there are some reasons why we think these teams exist, because of course they offer a lot of robustness. We have done this simulation, which I didn't have the time to go into detail today, but in this network, we deleted one edge at a time and then asked the question, is the phenotypic distribution changing or not? And when you do that 345 times out of 357, there was no change. Only 12 edges are actually making a change, and 12 divided by 360 is a small number, right? So there's huge redundancy which is present into the system. As I showed you earlier, that if you randomize the networks of lung cancer, you get anywhere between 10,000 to 1,000,000 states. But if you look at the wild type network, there were only 4 states. So clearly the biological networks are designed in a specific way that you get a limited number of phenotypes, not a whole conundrum of them. So these multi-stable dynamics can give rise to switching of states, can give rise to heterogeneity, which is not really at a genetic level. We did observe the existence of teams which can canalize, and we are currently working on how these landscapes can then be used to better understand how cells respond to different drugs. And hopefully these landscapes and these networks can serve as platforms where we can test out which combination of drugs is likely to be more effective as compared to which others. So of course there's more ongoing work in this. This is very new observations in our group. We are thinking about what are the necessary and sufficient features of a network to identify teams. Do all networks have teams? Which ones have, which ones don't have, and so on. We are also looking at, are teams seen only in cancers, or they're also seen in developmental biology setups? And where do we see them versus where do we not see them? This is another ongoing direction in the lab. And we are also thinking about, are there other advantages of teams? So we have 2 advantages so far, that there is robustness and there is canalization, but are there some other features also which are going on? So with that, I would like to thank the fantastic set of people I've had the fortune to work with in the lab over the past 4 years now. And I would like to thank the organizers once again, and thank you very much for your attention.
Questions?
In the Whitingstone model that you had, hello, am I audible?
No, I think it's.
Maybe you have to increase the volume. Yeah, so the Waddington model that we have shown that the cell takes a particular decision of cell fate. Yeah, so in the Waddington, the cells they take a particular decision for their I mean differentiation and all. However, using Yamanaka factors, it could be reprogrammed into that again. So is it really a dogmatic thing to say that it can go a unidirectional way, because using that thing we can go back as well? So that is my question.
Right. That's a very good question. So the question is that is Waddington landscape really one directional in nature, or can you push the cells from one to another? So yes, it was initially thought to be a very unidirectional case, but now there are enough instances of transdifferentiation. You can push one cell to become another, which of course Yamanaka factors are the perfect example of that. You can also push them back to so-called de-differentiation. So it's not unidirectional flow as was originally postulated. Having said that, this is unidirectional. The efficiency of reprogramming is still absolutely low. So the efficiency of Yamanaka factors induced reprogramming is still maybe 1 in 10,000 or 1 in 1,000 cells is able to become what they want it to become. So there are still open questions about why the efficiency is so low. So clearly there is some locking that is happening, but perhaps that locking is not as strong that it's irreversible. But in terms of reaction rates, k forward by k backward is of the order of 10 to the power 4, 10 to the power 5. So it's effectively still there.
Mohit, nice talk. I have no question about the transitions. Of course, transitions are taking place. My question is regarding a nomenclature of saying decision. Now, the way I understand, when the decision theory was developed by Abraham Wald and many others in statistics, and then spread throughout, there it was that you get information, and then you decide whether either decide or you wait and collect more information. So it is a continuous process of collecting more and more information, and then you decide at some point. Now here, the cell is, of course, the transition, for that you have the landscape and all that is fine. But when you call it a decision, how is it gradually collecting information? And if so, what is that information that it is collecting?
Right. So yes, that's a very good question. Can you really call these these transitions as decisions, and what information is being collected? So the decision making is a term which is mostly coming from developmental biology framework, that one cell which has the ability to give rise to so many of them at some point is deciding to go one way, and in most scenarios not really coming back because of various other factors that are also involved in the process. So the decision, the information that is being collected is the milieu of all signals, extracellular signals, growth factors, etc., that are present around a cell, which are triggering different cascade of reactions. And there have been some scenarios where it has been shown that if you give only one spike of one step function of a given growth factor to a cell, in that case the decision is not taken. It needs multiple such stochastic interactions for the decision to be logged and taken. So the short answer is that we don't know for every case what all information is being collected and what is the minimum amount of information that you need, both as an area under the curve or instantaneously, that would trigger a so-called decision or a transition.
It's a response, I have no problem.
Yes.
Yeah, but calling it a decision, that is where I have
Yes, I understand. Yes.
difficulty.
Right.
Particularly, it's a single cell, it's not
Yes.
an organism.
Right.
All right. Any other quick question? Yeah.
So maybe let's say like tree of decisions. Where is the temporal information coming in? Because like I think everyone can hear me anyway. So. Oh, okay. Yeah. So my question is that like it seems like at every bifurcation point, set of like two players are coming in and they are mutually inhibiting. So that's what like decides like which way to go, right? So where is the temporal information located? You know, like how far down the line it's going to differentiate in two different lines and then go on. So,
Right. I wish there was enough experimental data that could help us dissect that question. So at least for the first few stages, there is more experimental data coming up in terms of how those so-called decisions are taken. So the time information there is in terms of which pathways are getting activated at which time point. So before that, that entire cascade is off epigenetically or through various other interactions. But at some point, those, and again, it's not as discrete as projected, these are more continuous processes. Some of those pathways are getting above their threshold level to be called activated, and that's where the time information is present in the changing variables of the gene expression or epigenetic or whichever other variable that we are looking at.
All right, so let's thank Professor Mohit for a very interesting lecture.